Okay.
But Mr. Dehart will introduce Dr. Behe and give a little explanation of the format we'll be following this morning. So, Mr. Dehart.
Well, it's good to see you all on Friday morning. It's really a pleasure for us to have Dr. Behe here with us today. I mean, it's truly an honor for me. I remember the first time I saw him, I think, at a conference in Biola, and we had passed one another, and that's Dr. Behe. And so anyways, for me, it's like meeting, since a former baseball coach, it was kind of like meeting Mickey Mantle or somebody like that. So it's a pleasure, anyways, to have him here. Dr. Behe graduated from his PhD in 1978 from the University of Pennsylvania. He went on and did postdoc work at the NIH, and that's the National Institute for Health Sciences, in DNA. His specialty, his PhD dissertation, was on sickle cell anemia. And since then, he not only holds a position at Lehigh University in biochemistry, but he is also a senior fellow at the Discovery Institute. He has written 2 seminal books on intelligent design, and arguably one leg of that stool is Irreducible Complexity, which basically Dr. Behe coined. And his book in 1996, Darwin's Black Box, was one of the leading works that actually kicked off the intelligent design movement in a way. And since then, he has also written a book called The Edge of Evolution, which was published, I think, in 2007. And so anyways, it's just a real honor to have him here with us. He and I this morning are going to dialogue back and forth a little bit so that we break things up rather than just a straight speech. And we're going to go until about 9:45 here, and then at that time, we're going to open it up for questions. And hopefully during his talk, it will stimulate some of you to interact with him. You have a rare opportunity to ask Dr. Behe questions about what he's going to present today. So anyways, welcome. Yeah, it's good to have you here. We agreed that I sent out 4 questions to him in preference, and I think he has some slides prepared to answer those questions. Dr. Behe has kind of, the argument for the eye being designed is something that even Charles Darwin recognized at the time of his writing of The Origin of Species. But Dr. Behe, being a biochemist, not only looks at the eye as a whole and the parts that make it up, but he also looks at the chemistry of it. So it goes to another layering of complexity when you look at the biochemistry of vision and things. So Dr. Behe, could you, we said we would discuss maybe some other types of irreducibly complex systems. You have the flagellum and you have the eye, and we thought that, I think, could you share some of those with us?
Sure. I'd be happy to. I think the eye is a good place to start because it shows us the difference between what we knew about life, what we knew about biology in Darwin's time, and what we know about it now. In 1859, Charles Darwin, of course, proposed his theory of evolution by natural selection. And a lot of people were taken by it and saw that it could explain a lot of things in biology, but they thought that it couldn't explain some other things. It could explain some things, but not other things. And one of the things they wondered about was the eye, because it was known even back then that the vertebrate eye was a very complicated structure. It had a lens to focus light, and a retina to detect light, and it had tear ducts and ocular muscles and stuff. And people wondered if this idea of random changes in natural selection could put together something as complicated as that. And Darwin said he said in The Origin of Species, he says, yeah, it seems hard to understand how the eye could be put together. But he said, if you think about it a little bit more, maybe it could. Suppose we had an organism that had just a simple light-sensitive spot. Not really an eye, but something that could detect light. And an organism like that could tell whether it was in light or in shadow. And that would be helpful because it might need to get into light to get food. And if you took that light-sensitive spot and depressed the region in the organism where it was found, then light coming from one side might cast a shadow on parts of the light-sensitive spot, but the rest would be in light. And so the organism could tell what direction the light was coming from. And that would be a further improvement. And Darwin continued, if you took that depressed region, depressed it more and kind of curved it over and filled it with some transparent gelatinous material, well then maybe you got the beginnings of a crude lens, and that would be an even better improvement. So he tried to put together a scenario, not really evidence, but a scenario how something like the eye could come about by gradual changes that could be selected by natural selection. But he started with a light-sensitive spot. And that seems like a pretty specialized starting place. Now, what does it take to make a light-sensitive spot? This podium is not light-sensitive. The floor is not light-sensitive. What does it take to make a light-sensitive spot? Turns out that Darwin didn't even know. And in his book, he wrote the following. He says, "How a nerve comes to be sensitive to light hardly concerns us more than how life itself originated." Well, okay. And he was right to kind of pass over that question because the science of his time hadn't a clue how to address it. Just to remind you, back in those times, molecules were theoretical entities. Nobody was sure if they even existed. The cell was thought to be a little piece of jelly, protoplasm, not really anything very complicated. So they didn't have the tools, either the physical or the intellectual tools, to begin to address this question. Okay, well then we can ask, what has science learned since then? What does it take to make a light-sensitive spot? What happens when a photon of light impinges on your retina? Hold on to your hat. When a photon of light first hits your retina, it interacts with a small organic molecule called 11-cis-retinal. Now, 11-cis-retinal is a bent molecule like this, if you look at my arm here, like an elbow. But when the light hits it, it isomerizes and straightens out like that. And that's a switch. It's a switch that turns on the light detecting pathway. And many of the steps of that pathway are shown on the next slide here. Now, let me just say a couple things. First of all, I know it's a busy slide, so don't worry, I'll walk you through it. But the other thing is that this is not to the same scale as the last slide. And the retinal that we talked about is actually attached to this other molecule, which is labeled RH, which stands for rhodopsin. It's a protein that's found in the retina of an eye. And the retinal is about 1/3 of the length of this rhodopsin. But when the retinal changes shape, it forces a change in the shape of the rhodopsin to which it bound. When that happens, it exposes a binding site that allows it to interact specifically with another protein called transducin. When that transducin attaches to rhodopsin, a molecule called GDP falls off, and another molecule called GTP comes on. Now the rhodopsin transducin GTP complex acquires the ability to interact with another protein called phosphodiesterase, labeled PDE here. And when they bind together, this turns on the phosphodiesterase, which is a chemical scissors, which can cut a molecule called cyclic GMP, turning it into 5' GMP. Now there's a lot of 5' GMP in the cell, and some of it can be found over here by the cell membrane, and it binds to another protein, which is called an ion channel. The ion channel normally lets sodium and calcium ions into the cell. But when the cyclic GMP concentration decreases due to the actions of the phosphodiesterase, it falls off of the ion channel. That causes a change in the shape of the channel, which closes it down, and now these ions can no longer get into the cell. That causes a change in the voltage across the cell membrane, which causes a current to be sent down the optic nerve to the brain. And when it's interpreted by the brain, that is vision. So this is Darwin's simple light-sensitive spot. And I think you can tell that it's a little bit more complicated than he thought at the time. But we can ask, is something like this something that could be put together by the gradual steps that Darwin envisioned? And I think Darwin himself gave us a clue as to whether we could expect his mechanism to be able to put together something like this. He wrote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. But I can find out no such case," he said in 1859. Well, let's take Darwin at his word. What sort of a system or an organ wouldn't be able to be put together by numerous successive slight modifications? And Darwin always insisted on this, numerous successive slight modifications, a very, very gradual path of evolution, because he knew that if things improved too quickly or in very big leaps, then it would look suspiciously as if something other than random processes were involved. So he always insisted on this gradualism. Well, one sort of thing that could not be put together like this, it seems, is a system which is irreducibly complex, or has a property of irreducible complexity. And that just means you have a system or an organ or something that has a number of different components, and if you take away one of the components, the system breaks down. If we go back to the previous slide of the light sensing biochemical system, if we don't have the rhodopsin or the transducin or the cyclic GMP or the phosphodiesterase or the ion channel or any of a number of components, then the whole system breaks down. It's not like it works, less efficiently. It just doesn't work at all. And in my first book, Darwin's Black Box, I wrote about a number of different systems that science has discovered which have this property of irreducible complexity. And one that many people don't think about is the blood clotting system. You cut your finger and blood trickles out and it slows down eventually and stops and then heals over. It doesn't seem like a big deal. But scientific investigations over the past 50 years have shown that this seemingly simple process is extraordinarily complex. And it's put together by a large number of proteins. A score of different proteins are necessary for this system to work. Essentially, the protein that forms the clot can't be present as a clot, or else all of your blood would already be clotted and it would stop and you'd be in bad shape. So it exists as an inactive form called fibrinogen. And it has to be activated by another component called thrombin. But if thrombin were already active, it would then quickly activate the fibrinogen, and you'd kind of get frozen as all your blood clotted. So thrombin has to be in an inactive state called prothrombin. And that has to be activated by something else, which has to be activated by something else, and so on. So again, this seemingly simple process, upon closer examination, turns out to be enormously complex. And as I wrote in the book, I think it's irreducibly complex as well, and therefore a very big headache for Darwin's theory. So I went on for a little while, but I think that's the first question.
So, Dr. Behe, normally people think that evolution, we don't see it because it takes vast amounts of time for it to happen. And yet, in The Edge of Evolution, you write, it's not really the vast amounts of time. And you explain that through 3 different organisms that, you start with HIV, and you talk about Plasmodium falciparum, which is malaria, and you also talk about the E. coli experiment, Richard Lenski's. And so, could you explain why those 3 examples kind of fly in the face of this, that we have to have vast amounts of time in order to see evolution taking place?
Well, yeah, generally people who think about evolution, you think of fossils and the fossil record and a long time ago, and things changed gradually. But you can actually watch changes. Evolution is supposed to be just changes in DNA, the DNA of an organism, which helps the organism survive. And so the organism and its progeny will increase in the population compared to the organisms that don't have that helpful mutation. And scientists have investigated that in the past 30 years in ways that they hadn't been able to before, because new techniques had been invented to allow them to follow the evolution. And one particular experiment was carried out by a man named Richard Lenski at Michigan State University in the United States. And what he did was he took some bacteria called E. coli, and he grew them in a little flask in his lab, and they grew enough to fill the flask after 1 day. And after 1 day, he took a little bit of that and transferred it to a new flask with new media for it to eat. And it would grow there, and at the end of the day, he'd transfer that to a new flask. And the next day, a new one, and the next day, a new one. And he's been doing that for 30 years. And it turns out that over that 30 years, the bacteria have gone through 60,000 generations. And there have been trillions and trillions of bacteria that have occurred. And he's seen a number of beneficial mutations occur. And that they would occur and spread through the population. And so he's been able to watch evolution in action. But until about 10 years ago, he didn't know what the mutations were. He just saw that some bacteria improved and they grew faster and so on, but he couldn't tell what the changes at the DNA level were, because there weren't the techniques to be able to nail that down. But in the past 10 years, those have become available. And he went back and looked at them. And it turns out that most of the improvements, most of the beneficial mutations, were due to breaking genes. Breaking genes that were already there, throwing them out. And that allowed the bacteria to, say, grow faster. But it was not showing how complex systems could be put together. It turns out that many beneficial mutations are degradative mutations, ones that break down DNA that was already there. How could that be helpful, you could say? Well, here's an example. Suppose you had a car, you were driving your car, and you wanted to get better gas mileage. Getting better gas mileage would be beneficial. What could you do to quickly get a car that had better gas mileage? Well, one thing you might do is break off the side view mirrors. Because if you did that, there would no longer be air resistance as the car drove through along the road. And maybe the gas mileage would increase a little bit. Of course, you won't have your side view mirrors anymore, but maybe you can get along without them, or maybe you don't care about the other cars on the road. So that's an improvement in a sense, but it's an improvement by breaking something that was already there. And it turns out that Lenski's work was the first to show that this seems to be a main root of what we thought was evolution. And since then, it's become clear that it's a very, very frequent root. For example, here's a table of genetic changes, mutations that are helpful for people who live in malarious regions of the world to resist malaria. And the most famous one is the sickle cell mutation, which helps resist malaria, but it also can lead to sickle cell disease, which can cause death in a young age. So it's really not a very elegant solution. But there's a handful of other mutations too. What are they? Well, one is in a gene for a part of hemoglobin, and that leads to something called alpha thalassemia. And the mutation turns out to be to break the gene for that part of hemoglobin. Another is called beta thalassemia. The mutation breaks another part of the hemoglobin gene. Something called hereditary persistence of fetal hemoglobin. There are genes that turn off the kind of hemoglobin that a person has before birth. But in this mutation, those control regions are broken. And so the fetal hemoglobin is expressed into adulthood. And that seems to help with malaria. Another gene called glucose 6-phosphate dehydrogenase, degrade it or break it or throw it out. That helps with malaria. And other systems too. Here are some stories that have been, this is from Richard Lenski's work. And he reported that one of the most favorable mutations that he found was where something that is called the ribose operon, which has genes that code for something that makes the sugar ribose, were deleted. And yet that was beneficial. It was one of the most beneficial mutations he's come across. Here's to destroy the gene. Another gene helps to fight against heart attacks. It's called ApoC3. And the mutation that helps fight against heart attack is a mutation that destroys that gene. And it's not just humans. With horses, there's a mutation called the gatekeeper mutation that allows horses to trot more smoothly. And that's favored by horse breeders. And they select the horses that have that trait and breed them. And it turns out that it's a nonsense mutation, which means essentially it breaks the gene. And this came out just a couple months ago. The whole genome of the polar bear has been determined. And the polar bear is derived from the brown bear. And people were anxious to see what did it take to transform a brown bear into a polar bear. Whoops. Oops. And I should say, since I don't seem to have the slide, is that there were a number of different changes, and over half of them were predicted to degrade or destroy genes. In some circumstances that can help, but it does not explain where the complex systems came from in the first place that you can later break to make something else.
Yeah, that's great. And in most biology books, it's described as an arms race, right? Instead of a breaking of.
Yeah. I described it in Edge of Evolution as trench warfare rather than an arms race. People talk about arms races in the sense of getting bigger and better arms when two organisms have to compete against each other. But I wrote that it's more like trench warfare, where you have war and everything is starting to degrade, and you throw everything you've got at the enemy and you break everything you can if you think it can help.
You know, one of the hallmarks of design is this bacterial flagellum that you see around, and for a long time, right, you made the argument that it was irreducibly complex, and then there was a paper that was seems to be quoted often that says the flagellum, bacterial flagellum unspun. Could you just comment on that paper and your thoughts on it?
Sure. Yeah, I made this argument that I mentioned a few minutes ago on irreducible complexity, that it would be a big problem for Darwinian evolution. And an example of the system that I discussed in my book is a mousetrap, a mechanical mousetrap. A mechanical mousetrap has a number of parts: a spring, hammer, platform, and so on. And if you take away any one of these parts, it can't catch mice anymore. It's broken. So it's what I mean. That's an example of something I mean that is irreducibly complex. And I went on further to say that there are things like that, in that sense, at the molecular level of life. The cell is run literally by machines made out of molecules. And one good example is this guy here, which is called the bacterial flagellum, which is quite literally an outboard motor that cells can use to swim. And it's got a number of parts. This is the part that acts as the propeller. It spins round and around and around and pushes against water, and therefore pushes the cell through the water. And this is something called the hook region, which acts as a universal joint. It's attached to the drive shaft, which is attached to the motor, and so on. And I wrote that this is irreducibly complex too, because if you take away the motor, or you take away the hook, or the other parts of it, then it can't swim anymore. The bacterium can't swim anymore. Well, there's a man named Kenneth Miller, who's a professor of biology at Brown University in the United States. And he's an opponent of intelligent design. And he says, that's not true. He says, well, you can take away parts of the flagellum and it'll still function. There's an apparatus called the type 3 secretory system, which seems to be composed of things that include parts of the flagellum, and yet it has a function. It turns out it can pump proteins. It turns out the flagellum has to construct itself. And how it does it is that there's a hole in the middle of this drive shaft, and there's a pump down here, and it can pump proteins out here, and they add one onto the other to form this structure. I should add that this is just a little cartoon view of the flagellum, and it's actually composed of many, many more parts than this. But Professor Miller said that, well, you know, we took away parts and it still has a function. But he was confusing the issue. The point is not that can you take away parts and can you then find some other use for the things that are left over. The definition of irreducible complexity says if you've got a function for the system, and if you take away one of the parts of the system's function doesn't work anymore. And if you take away those other parts, then it no longer can act as an outboard motor. But Professor Miller tries to illustrate his point by attacking my example of the mousetrap. You know, is nothing sacred anymore. You know, this cute little mousetrap. He says that if you take away the holding bar, the bar that holds this hammer in place before the mouse comes along, why, he can use it as a tie clip. Well, that's fine, but it's still missing the point that you can't use it as a mousetrap anymore. You can't stabilize it to catch mice. And nonetheless, he continues to make the argument. And he's also said that why you can use parts of the mousetrap as a paperweight. or a clipboard, or a keychain, parts of the mousetrap. And I've pointed out, well, if you look at pictures of paperweights, or clipboards, or keychains, they don't look anything like a mousetrap. And if you had something like that, you would expect it to be a much different shape. I think that argument against irreducible complexity is a little strained. Okay, I'll stop it on that part there.
So recently, in your book, On the Edge of Evolution, you're trying to define the barrier, what evolution can actually accomplish and what it can't. And as you mentioned earlier, it can break systems and actually explain some things. You, I think, in your book mentioned that it could make 2 or 3 changes and still be able to function. And recently there was a paper that seemed to that was written that seemed to verify your results and your findings. And I don't know if you want to talk about that a little bit.
Oh, I sure do. Yeah. Thanks for bringing that up. That's very surprising that you would ask about that. Fortunately, I have some slides. One thing you want to do if you're a person like me is try to find examples of what evolution can do. Now, I'm happy to think that Darwinian processes can explain some things. They explain some things well, but I don't think they can explain the molecular machinery of the cell or very complicated systems. Well, where do you think, where's it reasonable to say that, random processes can explain this, but not much higher? Well, what you want to do to get a good limit is to find a system where there's evolution going on and there are enormous numbers of organisms, incredible numbers of organisms. And the reason you want that is because you want as many chances for random changes to throw up something useful as possible so that natural selection can grab onto it. And it turns out one of the best examples is, in fact, malaria. Now, you probably know about malaria, but let me just say that malaria is caused by a single-celled organism, which is transmitted by mosquito. And when a mosquito bites a person, the malaria goes in, hangs out in the liver for a while, and then bursts out and attaches to a red blood cell. And a single malarial cell invades a red blood cell and eats the hemoglobin inside. And it uses that material and the energy from it to make 20 copies of itself. And when 20 copies are made, it bursts open from the red blood cell, and each one of those 20 attaches to another red blood cell and goes in and makes 20 copies of itself. So then in the next round, you have 400 cells produced. And in the next round, 8,000, and so on. And in a short time, it can eat a lot of a person's blood. Well, and I showed you on a previous slide that there have been a number of mutations in humans that have tried to, that have been a little bit helpful against malaria. But in this past century, we've invented drugs and medicines to help battle malaria, for example. Well, the difference between resistance to atovaquone and resistance to chloroquine is that apparently with atovaquone, you only need to get one mutation to come up with resistance. But with chloroquine, you need to get 2 different mutations in a person's DNA at the same time to have resistance. Now, people have billions. There are 3 billion pieces, or things called nucleotides, the elements of DNA. So the odds of getting one change, one particular helpful change, is about 1 in several billion. Getting 2 separate specific changes are a billion times a billion or so, which would be 10 to the 18th. That's an astronomical number. And so I argued that if you need more than just one change, then Darwinian processes are very poor ones to help evolution along. So that one change, or maybe two at the most, is in fact the limit to evolution. And that doesn't look like it'll be enough to explain the complexity we found at the bottom of life. And some of my critics, and I should say that, in case you didn't know, I do have a couple critics on this. Like Professor Miller, didn't like my argument in The Edge of Evolution about that. And they said that, in fact, you probably don't need 2 mutations for chloroquine resistance. And a number of other folks said that as well. Here's reviews of my book in which people said something like that. But it turns out just a few months ago, a paper was published in the Proceedings of the National Academy of Sciences, which established that, in fact, 2 mutations are needed for chloroquine resistance. So I was like doing a little victory dance in my office over that one. So it was really not all that hard to see, but the critics, I think, just didn't want it to be true because they saw it would be a problem from them later on. But in fact, the result was solidly nailed down here.
So just in closing, and some of you hopefully are thinking of questions, Dr. Behe, what do you think is the future of ID and design arguments in general?
The future belongs to intelligent design. And it's not because of anything I'm doing or anything anybody else in the ID movement is doing. It's only because that's where the data is heading. The more and more we know about life, the more and more complex and sophisticated and elegant it is seen to be. The stuff that I wrote about in Darwin's black box, there are new systems which are even much more complex and irreducible and elegant than that. We are discovering systems, complex systems, that we didn't even know existed, and finding out that they're necessary for life. I just wanted to show a couple of neat little pictures here of things that have been discovered recently. It was discovered that this insect here, this good-looking insect here, in one stage of its life, it's called a leaf hopper insect, and it can really jump very high and very fast. And in order to do that, its back legs have to go at the same time to coordinate the power stroke. And it was discovered that there are actual gears, mechanical gears, that exist between the legs of this insect that allow it to hop and push at the same time on both legs. If Charles Darwin had seen something like this, I think he would have, run away screaming into the night. Another one is this. The one example that many critics of intelligent design always point to, they say that there are many bad designs in nature that no intelligent designer would make. And one of those is the retina, because they say it's wired backwards, that the nerves for the retina actually come out in front of the retina and then go back around it, so that there's a blind spot and so on. And they say that's a poor design. But recently, it's been discovered that the cells of the retina act as light tunnels, and they can funnel light down towards the retina, so that this phenomenon doesn't even exist, the one that they were worried about. And the author of this, who's no ID proponent, wrote that having the photoreceptors at the back of the retina is not a design constraint, it is a design feature. The idea that the vertebrate eye, like a traditional front illuminated camera, might have been improved somehow if it had been only able to orient its wiring behind the photoreceptor layer, like an octopus, cephalopod, is folly. So this traditional bad design argument has fallen apart. And so it's necessary to know that these types of arguments, saying, look at how this works, I can't think of any reason that it should be constructed like this, and therefore it's a bad design. Those types of arguments are arguments from ignorance. They depend critically on what we don't know. Do we know what the system is doing? Do we know how it's working? And as we know more, those things fall by the wayside. So I think that the argument for intelligent design is getting stronger and stronger just because we know more about life, more about biology, and that's where the data are pointing.
Well, that's it for my questions for Dr. Behe. Let's see, I'm not sure, Mr. Horn, do you have a mic? If any of you students would like to ask Dr. Behe a question, that would be a great time. You have time to actually interact with him.
So I had a question for Dr. Behe. You know who Dr. DeRosier is?
Yes.
So he did a specific research on the flagellum that you researched about, and also the Senia pasta, which is the bacteria that, that caused the bubonic plague.
Okay.
So from that, their design or their functions are highly similar. And for what Dr. DeRosier did was he compared them, the two components of these two bacteria, and then so the bubonic plague, it had the same substance of proteins, and however, the flagellum had much more proteins, so there were missing components found in the bubonic plague proteins. So, wouldn't this technically be a supporting point for natural selection on how it does have missing points? However, it is still functional for it was able to transmit. Transmitting disease to cells.
Sure, yeah, good. Yes, that's a good point. The point that he's making is similar to what Kenneth Miller made, and that is that this other apparatus which uses some components of the flagellum is called the type 3 secretory system, and it's used by some organisms, like the organism that causes bubonic plague, to inject proteins into other cells, poisonous proteins that'll break it down, and it kills the other cells and it eats them. Okay, so it helps it prey on other cells. The question is, well, isn't this a step towards getting a flagellum? And I say no, it's not, because let's review where we're at. Nobody knows where the type 3 secretory system came from. Nobody knows how it could give rise to a flagellum. A protein export system, like the type 3 system, has nothing to do with rotary motion. Okay, so we have a completely different function than the first one. So, in my view, what we've found is that the flagellum is much more sophisticated than we used to know. It's got not only the ability to rotate, but it's got within it this pump that helps it construct itself, which I didn't even consider in my first book. But it does not tell us, in my view, anything about how chance and selection could put it together.
I was just kind of curious about what your thought was on this, but I know that a lot of your critics have all examined your reasonings, and they've also known a lot of your they also know a lot of evidence for not natural stuff, for intelligent design. And I mean, in my opinion, I think that the intelligent design information that's backing it up is actually very sensible and very logical. And if you compare it to like the ones for evolution. But I was just kind of wondering why you think people remain as Darwinists or as evolutionists or as advocates for that, even though they face that.
Okay. Oh, because they're just stubborn. That's why. That's quite well. Why do I think that people resist this compelling logic that I have put together? I certainly agree with you that I think the development of science and biology has made intelligent design very compelling. It was a lot less compelling in Darwin's day when he thought cells were jelly and we knew a lot less than we do. So why did people stick with Darwin's theory? Well, it turns out not a surprisingly large fraction of biologists aren't sticking with Darwin's theory. They're skeptical of Darwin's theory and they're casting around for another explanation. But many don't like intelligent design, I think because of the philosophical and other ramifications of it. They just don't want to go there. Science in the past 100 or more years has gotten away and used itself as being able to explain everything in the whole world without remainder and would be unhappy if that were not the case. If you read the books that are published by university presses by leading biologists on evolution in the past 10 years, something like 100% of them say that there are big problems with Darwin's theory, and why don't we try something new? But invariably, what they propose just doesn't work, and that's because they're just trying to propose another unintelligent mechanism to explain what is, self-evidently a product of intelligence. When the subject becomes public, though, when evolution is talked about in public before general audiences, then, however, even those who are skeptical of Darwin's theory will, we say, circle the wagons, will become defensive and will protect Darwin's theory, just so these crazy intelligent design folks, don't get the satisfaction of being thought to have a point. But it's complex, and there's a lot of kind of intellectual drag on the idea that people are used to thinking of it this way for a long, long time, and it takes a while for new ways of thinking to spread through the scientific community. But the important point to me is that what does the evidence show, and the rest is just kind of sociology and politics and so on, and that's less interesting to me.
Okay, so my question is, well, first of all, I'm not doing good in biology. So I'm just saying I don't have like a good knowledge in biology and Okay. So my question is, if there's no intelligent design, like let's just say assume, what is the chances of just it appearing like that? Or is there to me, if it's saying intelligent design, since we have no explanation for it, for me it's like saying, I got 100 on my test, but I didn't study. That's like saying I used magic, but really there's no magic to me. Well, there could be, but my question is you know what I mean, right?
Sure. All right, yeah, that's a good question. Okay, so if there weren't intelligent design, then what would I think, how long do I think it would take to produce these things? And the short answer is never. I don't think they would ever be produced without intelligent design. Not only would you not build things up, but there would be processes that would tend to break everything down. The second point is an interesting one, and that is that what good is intelligent design? We're just saying that looks like they were designed. We're not saying who did it or how it was done or anything mechanistic about it. So what good is it? I think number one, it's good because it acknowledges where something came from. If you were out in the woods and you saw a big, or let's just make it more interesting. If you went to another planet, say you took a rocket ship and you were the first explorer on another planet, and you came across these fantastic machines that could, do fancy things, but you don't know how they worked. You didn't know who made them. Nonetheless, you would know they were designed, and it would save you a lot of effort to conclude they were designed rather than futilely trying to say, how did these machines arise by random natural processes? Another point is I think that acknowledging design will help us understand how things work in our world. And first of all, say, for example, in medicine, many evolutionists think that there are essentially no limits to evolution, so that scary viruses and bacteria and so on that cause diseases in humans will invariably overcome our drugs, develop resistance, and will be helpless before them. But somebody who thinks that these required intelligent design will think that there are limits to the ability of these viruses or bacteria to adapt. And we'll try to, say, discover drugs that are strong enough to stop, to be beyond their ability to mutate, have a better reason for looking for such drugs. And I don't think that exists in the other viewpoint. Yes, sir. Oh, I'm sorry. I'm overruled. Yes, ma'am.
Have other critiques like Ken Miller and Richard Dawkins respond to your argument for the bacterial flagellum and the chloroquine argument after it got verified?
Yeah, the flagellum, I think I've actually went through some of those in the talk. With the flagellum, that article that Mr. DeHart referenced about the flagellum unspun, and Ken Miller always thinks up cute titles for things, tried to argue that because the part of the flagellum could act as a pump, therefore irreducible complexity didn't exist. And I think I dealt with that in fact, then if you take away those parts, it can't act as a rotary motor, which was the function that we were focusing on. With other folks like Ken Miller and Richard Dawkins and several of those other people whose reviews of my book I kind of went through quickly towards the end of the talk. They've argued against my argument that chloroquine resistance requires a couple of mutations. But one of the nice things about science is that sometimes you can settle an argument by getting evidence. And people unconnected with this debate, who are just interested in dealing with malaria, a few months ago published a paper showing that in fact you do need 2 mutations in order for chloroquine resistance to occur. So they argue back against me, but I argue back against them, and that's the way it is when you're on a controversial subject.
I wanted to ask a question about, let's say that if Darwin were to be here right now, because Darwin's theory was, back long time ago, and because it was so compelling to all the people, it was legit to the people, that they would understand why it is. And right now, from your compelling arguments, I do think that it's understandable why you think that way. But I'm just curious, if there were somebody who to be actually making another theory, because theories are flawed, they're like ideas that are backed up by facts, right? So if there were to be a person who had a theory and an argument so compelling that it would actually break down both of these, would you have an argument that would actually back that up?
Well, if there was somebody who had a theory and evidence to back up their theory to show how something else could explain these, well then they'd have the evidence. But I can't argue against theories from the future. So a scientist can only argue about the information we have in our hand already, the experimental results, the data we have in hand, and interpret it in terms of what we know. And from what we know right now, unintelligent processes, not only Darwin's theory, but any other intelligent process of which we know of, that cannot explain these systems. And we know that when we see complex, interactive systems in any other context, we always know and always conclude that they were designed. And so the argument is that from what we know now, intelligent design is the best conclusion.
What makes me a bit incredulous about intelligent design proponents is that in several decades, the scientists don't really seem to have come up with any real insights about this designer or its mechanisms, as opposed to the scientific community that does not believe in a designer, which has been marked by continual inquiry and theorizing.
Okay, yeah, that's a good point. So it's been more than 20 years, and who is this designer after all? And how did this happen? And why haven't intelligent design proponents made progress on that? And that's a fair question, but you also a whole lot easier and a whole lot more straightforward than figuring out who the designer was or how the designer worked. Just for example, think of something like, the statues on Easter Island, or Stonehenge, or something like that. If you went and you saw those statues for the first time, you would know immediately that they were designed. But it took centuries before anybody could figure out how they could be made or who might have made them. The nice thing about concluding intelligent design is that you can come to a conclusion of design by looking at the system itself right there in front of you. You don't have to, do experiments or theorizing or anything. You can see it in what I call the purposeful arrangement of parts from the system itself. So that's fair enough, and I'm sure everybody would like to make progress on issues, but that doesn't affect the conclusion of design. Now the other thing is, that you said is that the scientific community, or those folks who don't conclude intelligent design, continue to search for answers, et cetera, et cetera. And that's great too, but you have to say, what have they come up with as they've searched? And one thing they've come up with is that paper that I showed here in the Proceedings of the National Academy of Sciences a couple months ago, showing that chloroquine resistance just requires a couple of mutations, and yet that second mutation makes it much, much, much more difficult. They've also come up with work that shows that the great majority of mutations that are helpful, beneficial, degrade systems, and so on. I'm not claiming that any of my work, any experiments or anything I've done, have led to a conclusion of design. I'm saying that the work done by that scientific community that you're thinking of, in the normal process of figuring out how biology works, has pointed strongly to design, and has also showed why Darwinian processes don't work.
So I understand that you believe in intelligent design, but I was wondering if there was enough evidence in the world that you would ever believe that evolution could also be a part of intelligent design, so such as animals adapting to their surroundings. That there's been many studies that animals can find ways to find food in a different environment, or such as human pigments can change from getting darker to lighter depending on sunlight. So could you ever believe that evolution can be also part of intelligent design, creating human race like things like that?
Yeah, sure. That's fine. So the question is, I think that, things could be set up so that new organisms could be made because there was a design in place that allowed them to change. Is that about correct?
So what I'm saying is that I believe there was a creator who created the world, but I also think that he didn't create everyone suddenly. So he didn't create a human like Mr. Horn, like he didn't create him like that. Like he created him he created him in like a kind of like a theistic evolution idea. Like he created him in a simpler form, and he.
Okay. Yeah, I think I understand what you're saying, but you have to realize what the crucial question is. The crucial question in this debate is not whether common descent is true, that some organisms gave rise to others, or that it happened slowly or suddenly or whatever. The question is, did it happen completely by chance, completely without any intelligent involvement, or did life require some intelligent, at least some intelligent input? And the moment that you say, well, yeah, some intelligent input,
or he, she, it, or whatever it is, created the entire universe, like every single thing.
I didn't say that. I'm a biochemist. I'm focusing on life.
So the intelligent designer created all the life.
Well, I am focusing on biochemical systems, okay? And I'm saying that these complex systems, sophisticated systems, required intelligent design. Yes. I don't want to make, I certainly think that the universe was created by God, and that, there's good reason to think it was designed, that is, made so that it would give rise to life. But I don't want to make my argument here overbroad. I just want to focus on these particular things.
Okay, and also in your presentation you mentioned random chance. So I just want to clarify, what did you exactly mean?
Random is something that happens as a byproduct of the laws of nature that nobody specifically intended. Nobody made sure that something would happen. So that, throwing some fair dice, the way that it would come up would be chance. If you load the dice, then it's not completely chance what comes up.
So, wait, let me. Okay, so you talk about random chance, right? So, if evolution and let's say natural selection was random chance, then why don't cats have wings? Since random chance might make them have wings. So, I just want to ask.
I have a picture of my cat that I have.
Okay.
Well, a Darwinist would say that cats don't have wings because it would not be beneficial to them. That having the wings would interfere with some of the other stuff they have to do, and therefore wouldn't be selected. The Darwinian theory has two parts: random changes, which can be bad, can be good, plus natural selection. And natural selection says, the ones that are bad, they just die out. The ones that are good help the organism survive and reproduce, and therefore increase in the population.
Oh, we have one last question for Dr. Behe.
Great. So I understand that one of the reasons a lot of scientists are hesitant to believe in intelligent design is because if you do, you have to, it does imply that there is something supernatural out there. So if you take this evidence to believe that there is intelligent design, there are implications of a supernatural being. So is proving that the end all, is that the goal of our science? Or once we have proved that there is intelligent design, are there further implications to us? Like, how do we deal with the fact that there is the existence of a God, or do we just leave it at that and say, look what science proves?
No, the goal of science is essentially to describe nature and see how it works and see how it got here to the best of its ability. So even if you think intelligent design is a good conclusion based on scientific data, that doesn't necessarily mean that the designer was God, although he certainly would be a good candidate for the role of designer. And it also does not say that science has to explain everything in our world, everything there is to explain. Philosophy, theology, and so on. Maybe they would then have a bigger role in explaining a lot about our world.
So, as you said, God would be one of the good candidates. I'm not sure, but I'm under the impression that you do believe that that was who the intelligent designer was. Am I correct?
I'm a Christian.
So how do you,
pretty typical.
once you've reached that conclusion, what are the implications for your life? Like,
For what?
What I'm saying is, like, this might sound attacking, but like, what's the point of proving intelligent design? Like, once we've gotten there, doesn't that mean we need to go further? And like, how would we know who that God is, or what we're supposed to do with the fact that we know something like that does exist?
Yeah, well, all of those things would take you beyond science. Okay, so at that point, I would wave goodbye, and you would go off and talk to investigate the natural world. You can look out and say, well, how did life arise? Did it arise by this one process or the other process or something? Well, it looks like it was intelligently designed. What good is it to know that? Well, because we want to know what our universe looks like. Science does. If you want to know about the philosophical implications, then you have to move beyond science.
You have a lot of good questions. I'm sure we could go on for a long time, but we have to go back to class. But anyways, please join me in thanking Dr. Behe.